Welcome to week 4 of BIOT 643. This week's lecture will be DNA recombination, cloning, and transfection. The outline for this week's lectures will be initially DNA recombination, where we'll discuss restriction enzymes in bacteria, how restriction enzymes are used in molecular biology, the insertion of recombinant DNA. The next major topic will be cloning, where we'll discuss the basic strategy, selection of desired recombinants using different forms of resistance such as antibiotic resistance, functional complementation, functional disruption, blue-white screening, library screening with southern blotting. We'll next discuss the insertion of the desired DNA sequences into vectors, and then also cloning with various vector types, expression vectors, shuttle vectors, and suicide vectors. Then we'll discuss transfection into eukaryotic hosts, the basic concepts, and vectors for transfection. And lastly, we'll discuss DNA libraries, the definition, the types, the features, and the construction. So let's start with restriction enzymes in bacteria. So generally we think of restriction enzymes as representing the bacterial immune system, protecting against viruses that can infect bacteria, bacteriophages, by cutting the foreign DNA. In the diagram shown here, we show that a phage is injecting DNA into the bacterium. The restriction endonuclease binds to the phage DNA, and the phage DNA is cleaved and inactivated. So how do bacteria protect their own DNA from digestion from these restriction enzymes? Well, it's done via methylation. And here we show that the bacterial DNA is not cleaved. The restriction endonucleases cannot bind to the restriction sites, and that here you see the bacterial DNA with the methyl groups because the recognition sequences have been methylated. So what is the naming convention for restriction enzymes? Here it just shows an example. The names tend to relate to the genus, the species, and the strain, and then numbered by the order of identification in the bacteria. So in this case of the Echo R1, the E comes from the genus, the Co comes from, like coli, the species, and the strain R for RY13, and 1 because it was the order, it was the first identified in that bacterium, so it's Echo R1. So how do restriction enzymes recognize sites on the DNA? Restriction enzymes generally recognize a specific sequence of DNA. These typically are palindromic sequences. So what does that exactly mean? Here we show the recognition sequence for Echo R1. And by palindromic, if you look at the 5' to 3' ends, it's GAATTC. And if you recognize, the G recognizes the C at the 3' end. The A is complementary to the A at the T at the 3 terminal end. The second A is complementary to the T on the 3' end. So this is what we term as a palindromic sequence. And if you notice on the lower strain, again from the 5 to the 3' end, you see the same GAATTC. So you have a palindromic sequence. And it reads from the 5' to the 3' ends on both chains. Here we show the sites for a number of other restriction enzymes such as HEN3, again, the palindromic sequence, the AAGCTT, or BAMH1, which is a GGATCC. So in all these cases, you have the restriction site, which is a palindromic site. You have the DNA fragments which are produced, and here we show the cohesive ends, or also called sticky ends, that are produced by HEN3 and BAM1. And these have overhangs which you can use to ligate your DNA sequences of other sources. You have some restriction enzymes such as ALU1, which generates blunt ends, i.e. there is no overhang of DNA sequences for ligating other DNA to it. So let's return to the concept of how do bacteria protect their own DNA from digestion. Here, just as we initially discussed, this is showing a section of DNA from the 5 to the 3' ends. Here highlighted in the darker blue is your restriction site. It's palindromic. And normally here your echo R1 will cleave between the G and the A on both strands. So that raises, how can this change and help a bacteria protect its own DNA? Again, the same sequence of DNA. In this case, if there is a methyl group placed on the A chain, now when the echo R1 comes, it is not capable of binding to the recognition site and thus does not cleave the DNA strands. Next, let's discuss restriction enzymes in molecular biology. There are two main uses for restriction enzymes in molecular biology. There is the restriction fragment length polymorphism, or RFLP analysis, and DNA digestion and recombination. First, the restriction fragment length polymorphism is the digestion of DNA with restriction enzymes that can be used for molecular typing. This is most often for the comparison between strains or isolates of the same species. For example, if we digest a 16,500 base pair linear DNA fragment with echo R1, this shows our linear DNA fragment. And when we run these on a gel, we see that we produce 3 fragments: an 8,500 fragment, a 5,000 fragment, and a 3,000 fragment. That tells us that there are 2 echo 1 recognition sites. If they were here, you could see that the 8,500 sequence would be first, the 5,000 would be second, and the 3,000 third. Or the recognition sites could be adjusted slightly such that we have a different set or different arrangement of these 3 fragments. Or, again, it could be such that the 5,000 is your initial fragment. Or another alternative. So these are all possible scenarios based on where the recognition sites could be cleaving the linear strand of DNA to result in these three fragments. So the question is, which one of or combination of these restriction endonuclease sites are the actual sites on these DNAs, and how do we determine that? Well, for more information, let's use a different restriction enzyme. We'll use BAM-H1. Again, the same strand of DNA. This again produces three fragments, which means there are two recognition sites. Again, the same way as we did previously with the echo R1, depending upon where these sites are located, we can end up with different size pieces of DNA. So again, the question is, where are those sites of DNA? So this is a single digest with echo R1, yields three fragments of 8,500, 5,000, 3,000. And the single digestion with BAM-H1 again yields three fragments, in this case of 9,500, 6,000, and 1,000. So what if we now do a double digestion with the echo R1 and the BAM-H1? We now end up with five fragments at 6,000, 4,000, 3,000, 2,500, and 1,000. So knowing that we have two recognition sites for each one of these restriction endonucleases, how can we figure out where these actual sites are? So if again we start with our 6,500, let's focus on the 3,000 molecular weight. It's present in both the double digestion and in the echo R1 digestion. And then if we look at the 8,500, in the double digestion, it's missing, which tells us then that there is a site for the BAM-H1 within that echo 1 derived peptide or fragment. Now if we look at the 5,000 from the echo R1 digestion, again it's missing from the double digest, which means again there must be a BAM-H1 site within that fragment. On the other side, there is a 1,000 fragment in both the BAM-H1 and the double enzyme treatment. There is also the 6,000 in both the BAM-H1 digestion and the double digestion. However, as we saw with the echo R1, in the BAM-H1 digestion, the 9,500 fragment is missing, suggesting again that there is an echo R1 within that fragment of DNA. And if we look at the development, there is a 4,000, a 3,000, and a 2,500 fragments in the double digestion of the DNA, So these must be being derived from the 9500, which means that two of the echo R1 sites are present in that fragment of DNA. So within the 8500, we see there is a 6000 and a 2500 fragment in the double digest, which means there must be, again, a site for the BAM H1 digest within that fragment of DNA. So again, there is a BAM H1 site in that section of DNA and two sites for the echo R1 within the 9500. For the 5000 in the echo R1, since it disappears, the only ones that fragments that account for that are the 4000 and the 1000, which means then that there is a BAM H1 digestion site within that 5000 fragment. So using the single echo R1 and BAM H1 digests, as well as the combination of the two, allows us to determine where in this intact section of linear DNA are our restriction endonuclease recognition sites. And therefore then we can now restructure that into this is the order in the original molecule. So by using these restriction endonucleases, we can determine the exact order in which each of those fragments represents from the 5 to the 3' end. And it allows us to identify where each of the endonuclease recognition sites are within this strand of DNA. So without even knowing the sequence, we can map the restriction orders of the peptides that are derived from our piece of DNA, and we can identify which endonucleases would be useful for allowing us to insert this DNA into various plasmids. Again, here it shows with our intact piece of DNA where our restriction enzyme endonuclease sites of recognition are. Again, like I said, because this is linear and each one generates 3 fragments, there are 2 recognition sites for in each restriction endonuclease. Now let's talk about the restriction fragment length polymorphism. Let's start with, if a mutation occurs in a restriction enzyme recognition site, the recognition site can be eliminated. Now here's an example. Here's our piece of double-stranded DNA, which has the echo R1 recognition site, the GAATTC site. When the echo R1 recognizes, binds, and cleaves. However, if that site is changed so that A now becomes a C, the site is no longer palindromic, and echo R1 endonuclease cannot recognize that site. Now it does not bind and does not cleave. So you have now eliminated the recognition site. On the other side, if a mutation changes a sequence into a restriction site recognition area, then a new enzyme cut can occur. Again, here's our piece of double-stranded DNA, and here you'll recognize that the GAATTC site has been changed to a GAATCC site by a mutation, such that now it is not an echo R1 recognition site, so it can't bind and it can't cleave. However, if a mutation occurs where that C is now converted to a T, you now have the echo R1 recognition site, the GAATTC site, such that the echo R1 can bind and cleave the section of DNA. So strains can be analyzed by RFLP to determine relatedness. And this can be used to identify spatiotemporal migration patterns. And this is an extremely valuable epidemiology tool. And here, for example, we show if you isolate 5 strains from 5 different locations, and you're interested in looking to see their relatedness and how they may have migrated from one site to another over time, this is an approach which would be very applicable. If you isolated your gene, you used the endonuclease and cleaved and generated fragments, you'll notice that from the isolate from site 1, you have 5 fragments of DNA. And site 2 has exactly the same 5 sites. So these are extremely related, and you could say they temporarily arose together. However, if you look at the DNA from site 3, we've lost an endonuclease site. So there has been a mutation over time such that now this gives a different pattern with only 4 fragments being generated by the same endonuclease. If we look at the DNA from site 4, again, we've lost 2 more sites and we've created a third. So now we have 3 sites, but mutations have occurred in 2 other sites where we've lost the recognition sites for the restriction enzyme, but we have gained a site for a restriction enzyme. And in site 5, we've retained the same restriction site that we gained in site 4, and we have gained an additional site giving us, again, 4 patterns, but 2 of them are new. So saying that site 5 was the last group to arise. So it gives us this sequential order of when these strains arrive from 1, 2, 3, 4, and 5. Now let's talk about restriction digestions. Restriction enzyme recognition sites can be used to cut out specific sections of DNA. In this example, we show a section of DNA which contains the genes for 4 genes, gene A, B, C, and D. So if, for example, we wanted to obtain gene B, in this case we're looking at this section of DNA potentially being cleaved with 3 different endonucleases: BGL2, BAMH1, and SAL1. So if we wanted to obtain gene B has recognition sites for BGL2 on both the 5' and 3' ends. So based on that, the optimal nuclease that we would use would be the BGL2. And as we see in this figure, while digestion ends up with 4 different fragments, There's one fragment which specifically contains our section which has gene B in it. In a similar fashion, if your goal was to isolate gene D, again, if you look at the intact piece of DNA, what you see is that on the 5' end, there is a site for SAL1, and at the 3' end, there's a BAMH1 site. So in this case, we'd have to use two different endonucleases. Again, that would yield 5 fragments, but one of those fragments would include the intact gene D. So how do we cut and paste these different fragments of DNA that we obtain? So here we show two different fragments of DNA. And if you'll note, these have our appropriate echo R1 restriction sites. They're palindromic. So we cleave them with the echo R1 restriction enzyme. And if you'll recall from earlier, in this particular cleavage with echo R1, we have sticky ends or overhanging sections of DNA. And it's important to note with the echo R1 that the two ends from these two different pieces of DNA are complementary. That allows us to anneal the two different sections of DNA fragments. So your pink portion of DNA will link to the blue portion of DNA. So again, it shows you a similar fashion. This is our section of DNA. It's been cleaved with echo R1. It generates fragments with complementary tails or sticky ends. These sticky ends that complement each other will bind. But if you note, there's one component missing. Even though these bind complementary, there is nothing which links these two pieces together. The annealing allows the recombinant DNA molecules to form by complementary base pairing. The two strands are not covalently bound as indicated by the shaded gaps in this diagram. So after the cleavage and the complementary binding, there's still some assembly required. If you recall, the formation of a covalent link between nucleotides requires the creation of a phosphodiesterase bond. This ligation step is catalyzed by DNA polymerase in replication. Here this shows the template strand and our nucleotide. You notice the hydroxyl on the 3' carbon will interact with the base, the phospho group, on the next base. You now create a phosphodiesterase bond. With the loss of a diphosphide. So if we go back to our original diagram, we have our two pieces of DNA. They're cleaved with the echo R1, forming fragments with complementary tails, and the annealing of the recombinant DNA molecules forming by complementary base pairs, but they're lacking the covalent bonds. However, with the DNA ligase, which we just showed forms the phosphodiesterase bonds, the DNA ligase seals the gap and covalently bonds the two strands of DNA. The next topic we're going to cover is the insertion of recombinant DNA. So bacterial competence, that is the ability of cells to take up free DNA from the surrounding environment. The evolutionary advantage of this is that it allows the incorporation of useful genes, such as the acquisition of antibiotic-resistant genes. Here we show an example. When a bacterial cell dies, it releases a fragment of its own DNA into the environment. This DNA fragment is taken up by a recipient cell, which incorporates the DNA into its chromosome. Many bacteria are not naturally competent. There are roughly 68 exceptions, which include the 4 that are shown here. Competence can be artificially induced in bacteria which are not naturally competent, and this can be achieved via electrocompetence and chemically induced competence. First, we'll present electrically induced competence. The cells are placed in a conductive solution in a cuvette with electrodes, and a brief electric voltage current is passed through the electrodes. It is thought that holes are punched in the cell membranes which allow free DNA to pass into the cells. And these holes will eventually seal. The second method of competence is chemically induced competence. Chemically induced competence is very similar to electrocompetence. Here we show a normal bacterium that is treated with calcium chloride. This then becomes a competent cell. A plasmid becomes bound to the cell exterior, and at 32 degrees, within about 2 minutes, the plasmid is transported into the cell, creating a transformed cell. Many commercially transformation kits contain cells chemically pre-treated into competence. In the laboratory, transformation is the artificial insertion of foreign DNA into a cell. There are two modes of this transformation. There is non-integrative. Here we show an example where a plasmid is taken up by a bacterium, and here we see the bacterial chromosome and the plasmids are separate within the bacterium. Even after cell division, these two sources of DNA remain separate. The second mode is integrative, and here we show that the plasmid is taken up by the bacteria and the chromosome incorporates the plasmid, and after cell division, this chromosome carrying the integrated plasmid is duplicated and divided into each cell. Next, we'll talk about homologous recombination. Homologous recombination is mediated by homology between DNA sequences on separate DNA strands. Here we show our targeted gene. The targeted gene is shown in purple and the mutated gene is shown in blue. And if you note, on either side of that particular gene are two pink segments that are homologous. Based on this homology, there is a double crossover recombination event, such that the mutated gene is now inserted into the area of the targeted gene based on the homologous portions on either side of the targeted gene. Homologous recombination can be used for insertion of desired DNA sequences. It can also be used for deletion of undesired DNA sequences. Here we show chromosomal DNA as well as your vector DNA. By homologous recombination, you see the vector DNA is now incorporated and splits the chromosomal DNA, and now the gene, this insertion of the vector DNA, causes a disruption in the original gene. Next, we'll present a very brief description of the basic strategy for cloning. Here we show the construction of a recombinant DNA molecule. We have a vector which is digested by a restriction endonuclease and a fragment of DNA. These are combined based on what we talked about earlier with the sticky ends, the complementary sticky ends of these two based on the digestion by the restriction enzymes. These are then ligated and added to a bacterium. The plasmid is then transported into the host cell such that you now have a bacterium carrying the recombinant DNA molecule. This involves what we also just discussed, the competence and transformation. You have multiplication of this recombinant DNA molecule. And as the host cell divides, you have the increasing number and multiplication of the recombinant DNA molecules within the bacteria. These bacteria can be then grown on plates so that you'll have bacterial colonies that are grown on solid media. And numerous cell divisions results in the formation of a clone. So why do we need to clone? Cloning is a large-scale amplification of inserted DNA. Here we show a single cell containing multiple copies of a recombinant DNA molecule. Then we grow this into a colony. This serves to produce several micrograms of recombinant DNA. Now this can be inoculated into a liquid broth and incubated for, say, 18 hours. This approach can provide up to several milligrams of your recombinant DNA. The important part of cloning, though, is that cloning can purify the desired sequences from your undesired sequences. Here we show three possible scenarios from your cloning. You can have a bacteria cell containing self-ligated vectors. You can have a bacterial cell containing the wrong recombinant DNA molecule, and what you really want is your bacterial cell which contains your desired DNA molecule. So why do we need to clone? While cloning is the large-cell amplification of DNA, each individual cell divides many times, producing a colony of the progeny. And each colony is a clone. And here we show an example diagrammatically and then a photograph of the actual kinds of colonies you would see. You have a petri dish with a solid media. These bacterial cells are grown. The cells divide and form colonies. And as you can see on the actual photograph of a plate, these individual colonies can be seen. So how do we select the desired recombinants? The gene of interest is actually small relative to the complete genome. So for example, in the E. coli K12, there are 4,639,221 base pairs. And if you look at that and say, this is the gene that I want to clone, for example, the TRPA, this is only 804 base pairs. That is approximately 0.017% of the total genome of the bacteria. So there are many more sequences that fall into the category of undesired. So how can we select or identify just one gene? So the problem is selection. So for example, if we now take our unligated vector molecules and we take all of the fragments that we just generated from our total genome and combine them, some of the vectors, because they have complementary sticky ends, will self-ligate. And so you'll end up with some bacteria containing self-ligated vectors. Some of these vectors will combine with your gene of interest, and so you will have the desired recombinant DNA molecule. So your bacterial cell will contain the desired DNA molecule. Unfortunately, the vast majority of these vectors will combine with the wrong recombinant DNA molecules. So you'll have bacteria containing the wrong recombinant DNA molecules. So we can now clone these onto a solid agar. And we form multiples of these three groups of clones. Thus the question becomes, how do we select only the clone with the desired characteristic or gene that we want? Well, first, there's direct selection. Direct selection is a manner in which there is a specific method you use to select only an environment where the correct recombinant can survive. This can be achieved by incorporating antibiotic resistance, functional complementation, functional disruption, or what is termed blue-white screening. The second method is library screening. Basically, clone identification by performing a clone library on each of your clones. And we'll discuss these more in detail. First, we'll talk about the selection of the desired recombinants using antibiotic resistance. The selection of antibiotic resistance uses an antibiotic-sensitive host for cloning. Transformation introduces an antibiotic-resistant gene on the insert or on the vector. Host cells that were not successfully transformed remain sensitive to the antibiotic. These are killed when grown on antibiotic-containing media. In contrast, host cells that were successfully transformed become resistant to the antibiotic, and they can survive when grown on antibiotic-containing media. So to select an inserted antibiotic-resistant gene, here's an example where we have plasmid R6-5, which contains a kanamycin-resistant gene. Digestion with echo R1 results in 13 different DNA fragments. Ligation gives 13 different recombinant DNA molecules when these are ligated into the echo R1 site of a plasmid. Only those plasmids which contain the resistance gene can grow on kanamycin containing agar. So only the recombinant carrying the CAN-R gene can survive. Selection of an inserted gene via antibiotic resistance on a vector. Here we show the RP4 vector, which contains the gene for kanamycin resistance, ampicillin resistance, and tetracycline resistance. When cells are grown, some will contain the RP4 plasmid and some do not. When grown in normal media, that is, media which contains no antibiotic, all cells grow. When grown in media containing tetracycline, only those cells which contain the RP4 plasmid can grow. Here, we show a few examples of some of the antibiotics commonly used as selective agents for successful transformation. So, the production of antibiotic-resistant genes. Antibiotic resistance is not due merely to the presence of the plasmid in the transformed cells. The resistant gene must also be expressed so that the enzyme that detoxifies the antibiotic is synthesized. It takes several minutes before the cell contains enough of the enzyme to withstand the toxic effects of the antibiotic. Here we show the plasmid, and if it contains the resistance gene and the proper conformation, it will begin to produce the antibiotic-resistant protein. The suggested transformation steps is a heat shock treatment, incubate in a non-selective media for 1 hour, and that gives time for protein synthesis to occur, and then plate onto a selective media containing the antibiotic. Next, let's discuss selection of the desired recombinants based on functional complementation or functional disruption. Selection via functional complementation restores a specific non-working function to a mutant host. It requires a host with its copy of the desired gene knocked out. It requires a desired gene that confers the selectable phenotype. And transformation introduces a working copy of that gene. The host cells that were not successfully transformed with the desired gene displayed the mutant or non-working phenotype. In contrast, host cells that were successfully transformed with the desired gene displayed the wild type or restored phenotype. An example is the direct selection for the TRPA gene cloned into a TRPA negative strain of E. coli. Here we show the treatment with the endonuclease, producing the fragments. And these fragments are then ligated into the plasmids, and one of those plasmids will contain your TRPA. These are now added to E. coli cells, which are TRPA negative. And those bacteria which contain the appropriate TRPA gene will express the TRPA protein. So when it is placed onto minimal media, only the TRPA recombinants can survive. The other side of the coin is selection via functional disruption. In this case, you disrupt a functional working gene in the host. Recombination introduces the insert into a selectable gene, visible or functional phenotype on the vector. Here we show the normal vector, which contains your gene product, which is a target gene for insertional inactivation. And the recombinant vector molecule, where the target gene is disrupted, so there is no gene product. In this manner, the host cells that were not successfully transformed display the phenotype, and the host cells that were successfully transformed no longer display the phenotype. For example, the vector PBR322 contains the ampicillin resistance and tetracycline resistant genes. These contain numerous restriction enzyme sites within the antibiotic resistant genes. A BAM-H1 resistant enzyme site within the tetracycline resistant cluster allows insertion to disrupt the genes. Here you see A shows the normal vector molecule, and B the recombinant molecule. In this case, we have the new DNA inserted in the BAM-H1 site. So in this case, we can select for antibiotic resistant disruption. So how do we select for cells that have lost antibiotic resistance? Simply, antibiotics kill them. And replicate plating. This gives you your handle, the disc. This gives you the ability to do replicate plating. This is your source plate. You stamp the source plate. You have components from those colonies adhere. You then place this on media without antibiotics and media with antibiotics. If you notice, of all of these, the media without antibiotics, all the clones grow. In the media without the antibiotic, you see the loss of a single clone. So in that manner, this allows you to identify the clone which has had the antibiotic resistance disrupted. You can then go back and select that clone and grow these so that you have the growth of the purified transformant. Next, let's talk about the selection of desired recombinants using what is termed as blue-white screening. Blue-white screening is a specific type of selection by functional disruption that utilizes a colored substrate for visual selection. It uses a vector with the LAC-Z gene for cloning. There is an insertion of the target sequence which disrupts the LAC-Z. The LAC-Z gene encodes for the protein beta-galactosidase, which breaks down lactose into simpler sugars. Here we show that lactose is broken down into galactose and glucose. Beta-galactosidase also breaks down X-gal. In this case, we use a 5-bromo 4-chloro 3-indole beta-galactoside. This is colorless. It gives rise to the beta-galactose and results in the release of bromochlorohydroxylindole, which is blue. So this selection by blue-white screening uses a vector with the LAC-Z gene for cloning. Transformation disrupts the LAC-Z. So here we show a plasmid with the intact LAC-Z gene allows the cells to metabolize X-gal to form blue colonies. This DNA to be cut the same restriction sites. And the recombinant plasmid cannot metabolize the X-gal and ends up forming white colonies. Here you see in the last figure we now have the DNA inserted such that it disrupts the LAC-Z gene. Here shows you a very vivid example. Plating on media with X-gal allows the visual selection of plasmids with an insert disrupting the LAC-Z. In the diagram we see blue colonies which contain the non-recombinants and the white colonies which contain the recombinants. The lower photograph shows you this in an actual colored photograph. So basically the blue colonies, in this case the beta-galactosidase, is synthesized normally, so you have your blue product, whereas the white colonies have the beta-galactosidase is not synthesized because the LAC-Z gene was disrupted by the insertion of your DNA. So there is no cleavage of your X-gal, so there is no blue color. Next, we'll talk about the selection of desired recombinants using library screening. Selection by library screening creates clones from each of the restriction fragments without selecting for a specific insert. Here again we show the plasmid, which is now being targeted and digested by the Echo R1, resulting in the 13 different fragments. These fragments are then cloned into plasmids. The resulting collection of clones containing all of the genetic material from the original genome is called a library. The library is then screened for the desired insert. This can be performed by hybridization, sequencing, or biochemical enzyme activity analysis. Next, we'll briefly cover the insertion of desired DNA sequences into vectors. Insertion via restriction enzyme sticky ends. Insertion of a fragment with sticky ends cut by a specific restriction enzyme requires a cut site for the same enzyme on the vector. Many commercial vectors simplify this by including a multiple cloning site, aka a polylinker. This is a sequence of DNA that consists of a series of recognition sites for many commonly used restriction enzymes. How about the case of the absence of restriction enzyme sticky ends? Such as, what about enzymes that don't leave sticky ends but leave blunt ends? Here we show an example of ALU1, which leaves blunt ends following its restriction enzyme activity. Sticky ends are not required, but greatly improve the efficiency of recombination. Various methods for attaching sticky ends to blunt end fragments exist. There are linkers, PCR primers with restriction enzyme sites, PCR primers with restriction enzyme extensions, and TA cloning. First, let's talk about sticky end additions. Linkers. Sticky ends can be attached to the blunted end fragments by attaching linkers. Here we show a typical linker which contains a BAM-H1 site. You have a blunt-ended molecule of DNA and you have a series of linkers. Use a DNA ligase. These linkers are attached to the 3' and 5' end of your blunt-ended molecule. Now, using the BAM-H1, you can cleave these linkers, and since the linkers have a BAM-H1 site which leaves sticky ends, your blunt-ended molecule now has sticky ends added to it. Another option for sticky end addition are PCR primers with restriction enzyme sites. PCR can be used as an alternative to restriction enzyme digestion of the entire source DNA. Here, this shows the earlier example of restriction enzyme digestion fragments derived from the whole DNA molecule of a bacteria. You can achieve the same thing using PCR, but now instead of having fragments, you have the PCR products. The next option is sticky end addition with the PCR primers with restriction enzyme sites. You can obtain a PCR product with a sticky end by use of a primer with a sequence that includes an internal restriction site. Here we show the primer sequence, which contains the palindromic BAM-H1 site. The resulting PCR product has incorporated this BAM-H1 site. Following BAM-H1 digestion, this PCR product now has a sticky end based on the BAM-H1 digestion. But what if the sequence of the primer will bind to does not include an internal restriction enzyme recognition site? Then the option would be a sticky end addition of PCR primers with a restriction enzyme extension. One can obtain a PCR product with a sticky end by use of a primer with a sequence that includes a restriction site located on an extension at the 5' end. Here we show an example of the primer located at the 5' end of the primer sequence, which contains the restriction site. In early PCR cycles, the extension hangs off the 3' end of the amplicon. In later PCR cycles, if it is fully amplified as part of the amplicon. So the next option should be sticky end addition with TA cloning. If you'll recall from PCR, you have your two chains. You add primers at the 3' end, and then you have the amplification. One more thing, many DNA polymerases, including Taq, have a non-template dependent activity that adds a single deoxyadenosine, an A, to the 3' end of the PCR products. And here we show that adenosine located on the 3' ends of both chains. This single nucleotide A overhang is a short sticky end. This short A sticky end can recombine with a complementary T overhang. And this provides a very easy way to clone PCR amplicons. This activity of Taq provides an alternative to add a sticky end to products of blunt end cut restriction enzymes. If there are free deoxy ATPs in the solution, Taq adds an A even if the true PCR is not occurring. Here again, with the Lu1, we show it naturally forms blunt ends. Incubating the restriction fragments with Taq at 72 degrees results in the addition of an A. At the 3' end. This can now attach to complementary T's on your vector. The next topic we'll discuss are vector types. Here are some examples of the commonly used vector systems. There are plasmids derived from E. coli, bacteriophage lambda, there are bacteriophage P1, bacterial artificial chromosomes derived from E. coli, yeast artificial chromosomes, and human artificial chromosomes. The interesting feature with some of these artificial chromosomes is these systems have an insert capacity of, in the case of the bacterial artificial, from 50 to 300 kilobases. In the yeast system, it can be 100 to 2,000 kilobase, and in the human artificial chromosome systems, greater than 2,000 kilobases. So what are the types of basic plasmid vectors? There are expression vectors. These are designed for high-level production of the protein encoded by the inserted DNA sequence. There are shuttle vectors. These are designed with two origins of replication, one for each of the two different hosts. And third, there are suicide vectors. These are designed without an origin of replication for the target host. Expression vectors are designed to express a foreign protein. Here we show the E. coli vector with the promoter, the ribosomal binding site and terminator with its unique restriction site. You insert a foreign gene into the unique restriction site. This now is used to transform the E. coli. Then this foreign gene is now expressed within the E. coli, which then can secrete the produced protein. Often, the high-efficiency T7 system is used for a high level of transcription. This requires the T7 RNA polymerase gene. The T7 RNA polymerase binds to the T7 promoter. And here we show this diagrammatically. And transcription by the T7 RNase polymerase is stopped by the T7 terminator. So we mentioned this origin of replication. So what exactly do we mean? The origin of replication, this is the synthesis of new DNA strands from the original template, begins at a region called the origin of replication, or ORI. Synthesis proceeds bidirectionally outward from the origin. The origins of replication can be recognized very specifically. This is often genus or even species dependent. Now let's talk about shuttle vectors. E. coli is used for routine molecular cloning procedures. However, often other organisms act as the final host, such as the gram-positive Bacillus subtilis or the Agarobacterium for plant transformation. It's designed with two origins of replication, one for each of the two different hosts. Here in this diagram we show the E. coli origin of replication and your target host origin of replication. Genes can be shuttled between hosts. Initial cloning steps are conducted with E. coli, and fully developed construct is transformed into the final host cell. And the origin allows it to be replicated in this host cell. So let's show the shuttle vector process flow. You have a bacterium. The bacterium is now carrying a recombinant DNA molecule. And you see replication of the recombinant DNA molecules within the bacterium. You then look at this recombinant DNA molecule and it's transferred to a second bacterium. Now you have a bacterium carrying this recombinant DNA. And you see replication of this recombinant DNA within the second bacterium. So let's recall homologous recombination. This is mediated by homology between DNA sequences on separate DNA strands. If you'll recall, You have a targeted gene and a mutated gene. The targeted gene is shown in purple. Your mutated gene is shown in blue. And there are homologous strains shown in the pink regions on the 3' and 5' ends of both the targeted gene and the mutated gene. Based on the homology between these two sequences on the two different strands, you have a double crossover recombination event. This allows for the mutation to be placed into the targeted gene. Now we'll discuss the suicide vectors. This is similar to a shuttle vector, but this is designed without an origin of replication for the target host. So in this case, we have the E. coli origin of replication. Typically, this is used for integration of foreign DNA directly into the host chromosome, not maintained as a plasmid. So again, in this diagram, we show the gene which is to be integrated. Fully developed construct is transformed into the final host cell. The insert can recombine with the host genome due to homology. Here we show in pink the regions of homology, just like we showed on the previous slide. The lack of host-specific origins prevents it from being replicated. Again, as we showed in the earlier one, with the shuttle vectors, there was a second origin of replication for the host cell, which does not exist in these suicide vectors. So post-division daughter cells have lost the plasmid. So again, this shows the flow diagram of the suicide vector process. You have your first strain of bacterium. You transform it with a recombinant DNA molecule. In this case, this is now a suicide vector. It then replicates this recombinant DNA within this initial bacterium. The recombinant DNA molecule is removed. It is then transferred to the second bacterium. Now you have the second bacterium carrying the recombinant DNA molecule. Now, as you'll recall, since there is no origin of replication, you cannot replicate the plasmid. However, the gene of interest, which is carried by this vector, is integrated into the bacterial genome, and the vector basically commits suicide. Now we'll talk about the basic concepts associated with transfection into eukaryotic hosts. So here are some key definitions. Transformation. This is the introduction of exogenous genetic material into bacterial cells. Malignant transformation. This is the acquisition of cancerous properties by a previously normal cell. And transfection. This is the insertion of exogenous genetic material into eukaryotic cells. Transfection is the process of deliberately introducing and expressing a particular nucleic acid sequence, i.e., a gene sequence, into a host cell. Say a wild-type cell can be transformed into a transfected mutated genes. Mutated cells can be transfected with wild-type genes. Or a completely foreign gene. Transfection assays can be used to study the modification of gene expression and silencing, and protein function. There are two types of transfection. Transient transfections. This is when the introduced DNA molecule is not inserted into the host genome. The DNA is eventually lost during mitosis. Transiently infected cells are assayed 48 to 72 hours post-transfection. There are also then stable transfections. In this case, the introduced DNA is inserted into and becomes part of the host genome. As a result, the gene is passed on to future generations during normal cell division. Cells that are stably transfected need to be selected for and separated from the transient or non-transfected cells. The stably transfected cells are normally selected within about 48 hours post-transfection. So here are some examples of transfection methods. Cationic molecules can interact with negatively charged DNA, and these are subsequently taken up by the cell via the process of endocytosis. There can be lipid-based spheres, or lipofection. Examples of these are calcium phosphate and cationic liposomes. There are other transfection methods, such as electroporation. In this case, the electric potential across the membrane drives the DNA across the membrane through pores. Charged ions and DNA flow through the pores, then the cell membrane discharges and the pores close, leaving the DNA molecules inside the cell. There are nanoparticles, which can be taken up by cells, and microinjection, where DNA can be injected directly through the cell membrane into the cells. So for transfection into eukaryotic hosts, what are the vectors for transfection? Vectors are a carrier used to introduce foreign DNA into a host cell in the cloning of DNA. Plasmids and viruses are commonly used as vectors. Vectors are capable of automatically replicating within the host cell. They are able to express the genes within the host cell. And plasmid vectors rely on cell division for the transfer of DNA into the nucleus. Plasmids can be modified to be used as vectors capable of transporting the desired DNA. Plasmids used in cloning applications have the following features: a small size, an origin of replication. They typically replicate in the target cell at high copy numbers. They need to have a selectable marker gene and multiple cloning sites. Plasmids are often amplified in bacteria and then used to transfect different types of cells, such as mammalian cells. Thus, each application required two distinct origins of replication, one unique to the bacteria and one unique to the mammalian cell. Certain origins of replication play roles in the generation of low or high copy numbers of plasmids. The origins that code for high copy number plasmids are preferred. They can influence greater stability of the plasmid, and more plasmids are produced in a shorter time. But they can have adverse effects on the metabolic growth rate. We also indicated one of the things they're required to have is a selectable marker or reporter gene. These are used to select the transfected cells and separate them from the non-transfected cells. Commonly used selectable markers include antibiotic-resistant genes, genes encoding visually selectable traits such as fluorescent proteins, and enzymes that catalyze colorimetric reactions such as the beta-galactosidase. Other plasmid features include plasmids may be genetically engineered to contain specific DNA sequences such as enhancer and promoter sequences, signal peptides, and polyadenylated sequences. First, enhancer and promoter sequences are regulatory sequences where transcriptional factors and regulatory proteins bind and control expression of the specific gene. Enhancer and promoter sequences of different genes may be specific to an organism and may be active only in specific tissues. Plasmids can be engineered with appropriate regulatory sequences that are inserted upstream of the gene of interest. That would be 5' to the restriction enzyme sites, specific to an organism and may be specific to a tissue. Signaling peptides are a short peptide sequence on every protein that use to interact with the transport system in the cell to direct the protein to the correct cellular location. Signal peptides are often cleaved off once the final destination has been reached. A plasmid may be engineered to carry a signal sequence to direct the protein product of the inserted gene to the specific location inside the cell or direct it to be secreted outside of the cell. So for these polyadenylated sequences, these are specific sequences recognized by the polyadenylation machinery, which adds a stretch of adenines to the tail at the 3' end of the RNA molecule, typically referred to as the poly A tail. RNA molecules that are polyadenylated are said to be mature and are more stable. These polyadenylated sequences may be inserted 3' to the cloning site to be used for polyadenylation of the RNA product. Here, we show a specific example of the pcDNA trademarked, the his-lac-Z vector, and some of the features that it contains. It has the, for example, the human cytomegalovirus immediate and early promoter enhancer. It has the T7 promoter priming site, an express epitope tag. It has multiple cloning sites and 3 RNA frames, for example. And it also has a number of origins of replication as well as multiple resistance genes to act as a signaling molecule, including amphotericin or ampicillin resistance genes. The next topic are DNA libraries. What is a DNA library? This is a collection of DNA fragments from an organism that can be cloned into any of the vectors mentioned in the earlier section. The choice of a vector is determined by the size of the inserted fragment. One can say that a DNA library is a repository of a large number of recombinants. So why do we need DNA libraries? Well, we need these to be able to express a protein of interest on a large scale. The gene encoding that protein has to be isolated, cloned into an appropriate expression vector, and then expressed as a protein. To fish out a gene from the genome of any organism, we need to establish a DNA library. So what's the difference between a genomic library and a cDNA library? Well, chopping up the entire DNA content of an organism, either mechanically or with restriction enzymes to generate small fragments. Each of the fragments is then cloned into a vector molecule to generate a recombinant. A large collection of these recombinant DNA molecules make up a genomic DNA library. Changing the DNA to messenger RNA by the process called transcription and using the mRNA population thus generated to make the library. The library generated in this manner is termed a cDNA or complementary DNA library. Called cDNA because the messenger RNA is used to generate a complementary strain of the DNA which is made double-stranded and is then cloned into a vector. So here shows an example of the sequence or scheme to generate a DNA library. Here we show the chromosomal DNA with 3 genes, and then these on the case on the left, these are digested with the restriction enzyme. It generates DNA fragments. This DNA is cloned into plasmids. These plasmids then constitute the genomic DNA clones or library. On the right, again, we show the same piece of DNA with the 3 genes. These are then transcribed and to create messenger RNA. Then by the process of reverse transcription, we create cDNA, and the cDNA is cloned into the vectors. This now establishes the cDNA clone library. So genomic library features are, they represent every gene essentially equally. They have overlapping clones. They have promoters, introns, etc. Most clones contain only part of the coding sequence of a gene. And a large insert genomic library is more likely to contain all of the genetic material within a few clones. In contrast, the features of a cDNA library is it represents individual genes. Not all genes are represented. There are no promoters or introns present. If the gene of interest is highly expressed in that particular tissue, then there will be an abundance of that messenger RNA. It is easy to isolate the gene of interest because it will be enriched in the cDNA library made from that particular tissue. So what are the types of cDNA libraries? There can be full-length or PCR mediated. In the case of the full-length library, in this library, each cDNA captures the entire length of the messenger RNA. In the PCR-mediated library, the starting material is total RNA for cDNA sequence. In this figure, we now show the conventional versus full-length library construction, where you have the full-length RNA with the poly A tail and your poly T primers used for development of the library, versus on the right where we have the introduction of a sequence tag into the full-length messenger RNA. And lastly, this figure presents a schematic of PCR-mediated library construction. In this case, you isolate all the messenger RNAs produced by a cell. You then, using reverse transcriptase, create a DNA hybrid. This undergoes hydrolysis, and then as you see at the bottom, you have your double-stranded DNA after one cycle of PCR. You undergo PCR amplification. Ultimately, these PCR products are then inserted into your vector. And for your reference, these are the references that may be helpful for the future.